Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Before we dive into this episode, we have a few announcements to make. We are thrilled to announce that our ecosystem is growing and are delighted to count Domain Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, and Montana Molecular as our ecosystem partners for the next year. Become an ecosystem member yourself and join our partners and your colleagues today. The ecosystem is your GPCR-focused virtual playground. Join over 600 of your peers who have already started exploring, connecting, and collaborating better. You can explore the ecosystem by signing up and getting a free site membership. For the next couple of weeks, once you sign up for the free account, you can watch the talks from the third edition of the Dr. GPCR Summit for free. When you're ready, you can also get a premium membership to unlock the ecosystem's full benefits. If you'd like to register your team or your company, or if you live in a developing country, please reach out to us by email or in the ecosystem, and we'll be happy to help you join us at an adapted premium membership fee. The list of benefits of the ecosystem is quite long. Today, we want to highlight that you can get direct access to our ecosystem partners, ask questions, and who knows, maybe find your next job directly in the ecosystem. We also have a job board. It's a GPCR-focused one where you can explore different opportunities. And if you're looking to hire, you can submit your own job description. Wondering what GPCR meeting to attend next? Well, check out our events page where we have curated the next GPCR meetings for you. In case you're organizing a meeting, fill out the event submission form and advertise your event in the ecosystem directly. Take advantage of everything that the new GPCR dedicated online playground has to offer today. Explore the possibilities by navigating the site using the direct links in the footer. The footer is your best friend. Check it out today at drgpcr.com. And now, let's dive into this episode. Hello, everyone. This is Yamina from Dr. GPCR, and I'm very excited today to have a new guest on the podcast, Dr. Robert Laprairie. I'm so excited to have you on, and my French accent, my French played in introducing you. Hi, Robert. Nice to see you tonight.
Nice to see you as well, and great to be talking to you.
I'm very excited to talk to you. So let's start at the beginning. Why don't you walk us through a little bit, introduce us to who you are and how you got to where you are today?
Yeah, I'd love to. So I am the Associate Professor and Research Chair in Drug Discovery and Development at the University of Saskatchewan in Saskatoon, which is right in the middle of North America, if you're not familiar. I got started in research as a research summer student at Agriculture Canada, actually working on canola controls for pesticides. And then transferred from there into starting some pharmacology work, some cell signaling work, looking at protein phosphatases in colorectal cancer models. And then from there, that's really after that when my interest in GPCRs kicked off. I started a master's with Eileen Donovan-Wright at Dalhousie University, looking at the type 1 cannabinoid receptor in the context of Huntington's disease. Finished a master's with her, and then after that master's stayed on as a PhD student and really focused more on CB1 molecular pharmacology. Did a brief postdoctoral fellowship at the Scripps Research Institute with Dr. Bond, and then after that was able to get the current faculty position I have, where I've been for the last 6 years at the University of Saskatchewan. And now we're primarily focused on the cannabinoid receptors, but we do a little bit of work on orexin receptors and 5-HT2A serotonin receptors too. So just all kinds of GPCRs.
I love that. That's so interesting. We have two things in common. I'm also from Canada, as I mentioned before we hit record. And the other thing is that you completed your master's and PhD in the same lab, and I did the same thing. But we're going to get back to that. You mentioned, starting out working at Agriculture Canada. What got you interested in science in the first place?
Yeah, so I've always loved science and research as a kid. I was definitely, the nerdy kid in class that was interested from that perspective. But I saw myself becoming like a high school science teacher, and that was kind of the trajectory that I was on. But early on in my undergrad, a buddy of mine, he had been working at Agriculture Canada, and he got into medicine, and he said, Robert, we need someone to take my job at Agriculture Canada. It's conveniently located job on campus, and it's a research job. And I took it honestly thinking like this is good money. It's close by. It's convenient. But I ended up loving it. I loved the investigation of it. I enjoyed the lab culture and talking through problems and experimental design with my lab members. And yeah, I just kind of thought I changed my career trajectory after that to focus on what didn't feel like work.
Yes, and I think you make a great point about, trying out something and ending up, really loving it. And the other point is doing something that doesn't feel like work. And I think that's very important. We'll talk a little bit later about advice for junior scientists, and I think that goes into that category. But first, let me ask you, do you remember the time, the moment when you first heard about GPCRs?
Yeah, so it would have been in a fourth year cell signaling course. So I was in my fourth year of my biochem degree, and we were talking about GPCRs in the context of protein phosphorylation and signaling.
Mhm.
And so I remember one of my professors saying, 3 out of every 4 drugs or something like that approved by the FDA targets this type of receptor, and just being kind of blown away by that, and all of the ground that had been covered, all of the biology that's covered by a pretty uniform mechanism is amazing.
That's pretty much what I like. That's the intro slide to any
Yeah,
GPCR
yeah.
talk you might go
Yeah.
to. And if you go to a conference, there's 20 talks, and you can be sure that at least 15 out of those talks are going to say, 30 to 40% of FDA approved drugs target GPCRs.
Yeah.
And what fascinated you about GPCRs in general? Was it the aspect that most drugs, or a large number of drugs that are FDA approved, target this family of receptors? Or was there anything specific that you remember telling yourself, this is interesting?
So for me, it was actually more about the cannabinoid receptors than GPCRs in general. And then I
Mhm.
came to appreciate the unified mechanisms later on in scientific life, I would say. So initially, I was drawn in to work on CB1. And for CB1, there's this weird kind of lag compared to a lot of other GPCRs or GPCR dependent signaling, where the state of the art, at least for a time, was probably about 15 years behind comparable GPCRs like the adrenergic receptors or the
Yeah.
muscarinic receptors. And so it just seemed like there was this huge void in terms of our understanding. But there had been all these great tools developed for other GPCRs that could easily be built on into the cannabinoid receptors and used to study this system, and gain a lot of ground very quickly. So I think that's what brought me in, or what hooked me.
What at the time when you were thinking about, these tools that were not applied to the cannabinoid system,
Hmm.
um, what was the first thing you thought about? You told yourself, well, there's all these tools that are used for the beta adrenergic. I think it's interesting. I'm gonna open a parenthesis here. It's interesting because everybody says, well, you want to have a good control, use the beta adrenergic receptors,
Sure.
which is great. And
Uh-huh.
we have all these tools and all these tool compounds. But what I'm trying to say is that we still don't know enough about even the beta adrenergic receptors. There's
Hmm.
publications that come out that reveal new things, new discoveries around these receptors. But
Yeah.
when you're thinking about the cannabinoid receptors and the tools that were available for model receptors like the beta adrenergic, what was the what drove you? Like, how did you pick the first tool or the first application to test with the cannabinoid receptors that was already available for the beta adrenergic, for example?
Yeah. One of the things that I really love about the cannabinoid receptors, or really any GPCR, is just the opportunity to ask a really simple, directed question. So, for example, with CB1 or CB2, what is the time course for beta arrestin interacting with this receptor?
Mhm.
Right? And so there were, for example, plasmids or tools that had been developed that allowed you to look using something like bioluminescence resonance energy transfer, or BRET, and just say, does beta arrestin even bind to CB1? And, that in itself is an important question. And then from there, kind of spilling out into the other questions of different isoforms or time dependence or ligand dependence. I don't know if I fully answered your question. I kind of got carried
Well,
away there.
I think you kind of you did, in the sense that there were so many unknowns at the
Hmm.
time in the CB1 or in the CB realm,
Yeah.
independently of which receptor you're looking at, that anything you asked using the tools that were initially developed for the more studied receptors was allowing the field to better understand the cannabinoid system.
Yeah, absolutely. And I think it's great when you can ask those pointed or open ended questions that are very directed and still get useful information. I think in a lot of fields outside of GPCRs, sometimes it can feel like you have to ask a very niche question in order to grow the field, but I don't ever feel like that's the case for
Mhm.
GPCR research.
And so, and this was your project during your master's and your PhD?
Yeah.
Wow.
So during my master's, it was very focused on CB1 in the context of Huntington's disease.
Mhm.
In that disease, we see a precipitous loss in CB1 expression in specific regions of the brain that are involved in the pathophysiology of the disease. And then for my PhD, I wanted to broaden that out a little bit more and ask some more basic questions about the signaling of the receptor just in a normal state,
Yeah.
in air quotes, because
Yeah.
that's, yeah.
It's so interesting. You know, you mentioned Huntington's disease, and my brain goes directly to the mGluR receptors.
Mhm, mhm.
Um, do we know, and totally naive question, and maybe I should know this, do we know if the mGluR system interacts with the cannabinoid system in Huntington's, or in normal states or natural state, and does that change anything? Is there any change in the Huntington's disease setting?
Yeah, so, you know what? I don't know, quite frankly, either, whether there's been any evidence for direct interactions between mGluRs and cannabinoid receptors. I do know that there are some mutations in mGluR2 and 3, I believe, that are considered disease-modifying mutations for
Mhm.
Huntington's disease. So certain individuals can have a worse case prognosis if they have these
Yeah.
disease-modifying genes. But beyond that, I'm not sure if there's been any study on the interactions between those receptor systems.
I'm wondering even if there is any functional interaction, because to
Yeah.
be frank, I think there is so much work to be done, not only
Mhm.
in the cannabinoid system, but really looking at GPCRs individually
Yeah.
from a molecular pharmacology perspective. But then I think the next level of this would be to look at the cross interactions functionally
Mhm.
of these GPCRs in a given cell system, in a given organ, in a given disease. I do understand that we don't have necessarily the tools, but even having some kind of bioinformatic tools to
Right.
even compile what we call the basic, and I'm also using air quotes, the basic molecular pharmacology, and see if there's any, functional potentially interactions, which I think is really fascinating.
I do too, and I'll jump around a little bit in the timeline, I guess, but one of the people that I was doing my PhD alongside, Amina Bagger, she was looking at interactions between CB1, CB2, and dopamine D2 receptors. For the same kind of idea of what physical interactions exist, what could be going on there. And now in my own lab, I have a student that's interested in orexin receptor interactions, or some people call it hypocretin interactions with cannabinoid receptors in the hypothalamus, and
Mhm.
how that affects stress response and feeding behavior.
Interesting.
Yeah.
Interesting. So do you have a favorite GPCR? And we're gonna get back to the science in a minute, but I think kind of the answer is kind of obvious, but still I'm
Uh-huh.
gonna ask it.
So CB1 is my favorite GPCR. It's the one that I've studied the longest. I think
Mhm.
it's fascinating. And I one thing I really like about CB1 is, its primary ligands are all lipids. And that makes it kind of weird because there's still a bit of mystery around, how do these ligands get there if they're not water soluble and what's
Yeah.
what's going on there? Yeah. Yeah.
I think cannabinoid receptors are also really interesting, from multiple perspectives. The fact that you just mentioned that they bind lipids, and also the fact that there's natural and natural in the human system and
Mhm.
also natural plant, natural type of ligands
Yeah.
that will activate the cannabinoid system. You
Mhm.
mentioned, so what do we know? What is the latest and the hot off the press around the cannabinoid system that we didn't know, for example, when you were doing your master's?
So I'd say the kind of the latest for the cannabinoid receptors are there's two different things going on that are kind of the latest and greatest. One has to do with allosteric pharmacology,
Mhm.
and I'm maybe a little bit biased here because there's a lot that we try to do in this area, but a lot of work being done to try and figure out where positive allosteric modulators might help or sit on the receptor that might augment signaling. And then some really interesting work on synthetic negative allosteric modulators coming from a couple of groups. And I'll just highlight Ruth Ross from the University of Toronto, looking at really interesting negative allosteric modulators or NAMs in the context of regulating things like appetite or substance use disorder that I think are really fascinating. Um,
Interesting. And in
yeah.
what context would you be looking at a positive allosteric modulator?
So we've been exploring it in the context of certain types of epilepsy and seizure, primarily in rodent models of absence epilepsy.
Mhm.
And then we're collaborating with a group out of the University of Sydney to look at other models, rodent models of seizure. And then Andrea Homan is using the same, so she's at Indiana University, using the same for pain and specifically chemotherapy induced neuropathies.
Interesting.
Yeah,
So much work to be done.
yeah,
Let's go back into the timeline.
sure.
You're finishing your PhD. Why did you decide to do a postdoc? I remember that you in the beginning you had mentioned that you were getting ready to become a high school science teacher, then you
Mhm.
got to know what science and research is and decided to stick to it, but what made you jump and think about being a professor?
Yeah, so I think for better or for worse, I felt like, if I want to be a professor eventually, I want to run my own group. I want to supervise and train younger scientists. The next logical step is to do a postdoctoral fellowship.
Mhm.
So let's go and do that, and then move through that into a faculty position. And so it was kind of like, I guess you could say in a way that I had blinders on in a bit because I thought like the only way to faculty position is postdoc.
Postdoc.
Um, Interestingly, I've now personally graduated two PhD students, both of whom got faculty positions without postdocs.
I think that's pretty rare,
Yeah,
which is I think it's awesome that that happened to two of your students.
yeah,
I think it's important to keep in mind that, a postdoc is not an answer to everything.
hmm,
And I think this pretty much goes back to I've spoken to a lot of people, and I think first thing that people have to do is figure out, okay, what do you want? What's my
right,
end goal?
yeah,
And then work back the steps and
hmm,
not automatically assume that there was a postdoc in your future. That
right,
being said, I did two postdocs.
hmm,
Um, the first one was because I really loved what I was doing in the lab. I did a lot of conformational function studies on chemokine receptors using bread, and I really wanted to move to New York. And I found this awesome lab, and I was super
hmm,
excited about it. The
yeah,
second one was an opportunity to learn something new, but
mhm,
I don't know if I were able to go back in time. I don't know if I would do it again,
that's
knowing what
fair.
I know now.
Yeah, yeah,
I'm pointing it out for the audience, especially for the trainees, just to get them to think about, okay, what do I want to do? And is a postdoc something I need to do or something I want to do? And no
right.
matter what the decision is, it doesn't matter as long as you jump with both feet into the opportunity, knowing what the end goal is.
Completely agree. Yeah.
How did you pick your postdoc lab?
I wanted something that was gonna allow me to learn new techniques,
Mhm.
and I wanted for my family to have a place to go where I felt like my family would also be able to thrive. So I have three kids. At the time, I was married. I'm now divorced, um, but I needed somewhere for my family to be able to
Yeah.
thrive. Yeah.
Which is again, I think it's a great. And thanks for sharing that. I think it's also a great point to put something to point out for the trainees listening is that it's especially if you have a family, which
Hmm.
most likely when you towards the end of your PhD, you're most likely at least have a partner, maybe
Yep.
some children, maybe some pets.
Yep.
But you have to think about that as well, and that's I think complicates, but at the same time helps you fine tune the decision making process.
Yeah. It lets you know where your priorities are, I think.
I think so too. I think so too. So, what made you pick Dr. Laura Bond's lab? What did you like
So
about it?
yeah, yeah. So we seem to be interested in many of the same things.
Hmm.
We wanted to explore some interesting things related to cannabinoids.
Hmm.
Um, and I think I knew other folks that were going to that lab, and it looked like it was going to be a really positive training experience for me.
And there's
Yeah.
also the weather to consider when you're
I
when
mean,
you're
I'm
leaving.
back in Saskatchewan now, so I don't know if I learned that lesson.
I don't know, but I think you kind of got a taste of
Hmm.
the Florida weather compared to in Canada in general. But I want to say Saskatoon. I mean, it's just I'm from Montreal, and I
Yeah.
think Montreal is pretty cold.
Hmm.
I remember watching the news and watching the weather forecast and looking at the temperature across the country, and there
Hmm.
was a there's a colder region which was kind of the mid region of Canada, and you're
Yeah.
in it
Oh yeah.
completely. You
Yeah.
also mentioned that your a postdoc was a short one. Was
Yeah.
that planned out, or was that because you were lucky and you were able to wrap up your project fairly
Yeah,
quickly?
so I would say that I've been incredibly fortunate. Um, a position became available at the University of Saskatchewan that really fit kind of what I was looking for. So
Mhm.
when I was initially hired, it was an endowed research chair focused on drug discovery and development. And the I think the key fortunate thing for me is the University of Saskatchewan was keen on finding someone that wanted to put down roots in Saskatchewan. And I was born here, so I like to come back home made sense for me. So I think, fortune had a lot to do with it, and I'm grateful for that. But yeah, yeah,
And that's why we understand why, you left sunny Florida to come back
mhm,
home.
yeah,
And when you grow up in a place, I mean, the cold is relative. I
absolutely.
it just and let's be honest. Yes, it's cold in Canada, and yes, it was cold in Montreal. But
Mhm.
every time you looked outside and it was sunny and everything is white and it's calm and it's beautiful, it's just such a majestic, you know, area to look at, and you're like, wow, this is actually quite cool.
Yeah, and I like the sound of snow crunching under my feet. I think it's just really nice.
I think so too. I think so too. I like it when it's snow, after the snowstorm when there's snow everywhere and
Mhm.
you're outside, but there's so much snow that you feel like, it's the difference in sounds between an apartment that's filled with with stuff versus an empty apartment. And that's how it felt being outside in the snow.
Yeah, yeah.
So coming back to Canada, in the department that you're in and the
Mhm.
teams that are around you, how does that look like in general? What do you do during the day? You mentioned having many students who work on different
Mhm.
projects.
Yeah.
um, can you tell us a little bit more about that?
Yeah, I'd love to. So my kind of day to day right now, like I said, I'm an associate professor, so I just got tenure this summer, and
Congratulations.
thank you. And right now we have a lab of about 8 people, ranging from postdoctoral fellow, PhD students, master's students, undergrad students. Fluctuates kind of depending on how many undergrad students are coming through the lab.
Mhm.
And so I try to devote my mornings to meeting with individual students, try to meet with everybody at least once a week for an
Mhm.
hour, talk about their research, talk about how it's going, help them if they're having trouble, these sorts of things. And then usually a weekly group meeting where we, talk about how life is going for everyone, maybe some things aren't working, troubleshoot as a team. And really try and, foster that team environment among the group. I'm again fortunate because my position as a research chair means that I only have to teach 20% of the time. which is fine with me. I really enjoy teaching, but I get to kind of pick which courses I
That's
teach.
awesome.
Yeah. And then this past, I guess, since the middle of COVID, I've begun a role as a co-chair for our university animal ethics committee. And so for that, twice a month, we meet, we review all of the new and renewed animal protocols, and something I'm really passionate about as animal ethics and research and responsible use of animals. So I've been loving that. And the University of Saskatchewan has a great animal care program that I just enjoy being a part of. It really stretches my thinking beyond GPCRs.
That's great. That's great. You come back to the lab refreshed, and now you can
Yeah.
dive back into the GPCR
Yeah.
research. What I also wanted to ask is it, since I don't know how big the University of Saskatchewan is,
Sure.
uh, to be honest, again, don't take it the wrong way, but in my mind, in the middle of Canada is the prairies, and there's a lot of snow and it's cold. And I
Uh-huh.
also know that Saskatchewan doesn't change the time, if I
That's true.
understand, which
Yeah.
is great because we're switching time over the weekend here,
Yeah.
and it's always a messy thing. And when I first heard that Saskatchewan doesn't do it, I said to myself, well, maybe we should do the same.
Hmm.
But how does the university? How big is the university? How easy is it for you to, for example, recruit students?
Yeah, that's a great question. So the University of Saskatchewan is about 20,000 people. It's quite it's a decently sized university. I'm in the College of Pharmacy and Nutrition, which
Hmm.
is a non-departmentalized, smaller college. But I love it because I get great support from my administrative team. Like my deans and my associate deans are all super responsive. And we're in kind of an insular, smaller community, which is helpful and supportive on all fronts. We have great access to kind of all the toys that you would expect at any major research intensive university. But because we're a comparatively smaller institution, I never have to wait for anything. And my students never have to wait for anything. So if one of my students needs access to a confocal microscope, there it is. Go, get on it. If another one of my students needs to do mass spectroscopy to measure the amount of a cannabinoid in the plasma of their rodents, there it is. Go, get it done. And so having access to all the tools with none of the wait time is pretty excellent. On the flip side, you asked about recruitment. Recruitment is tough, you know.
Hmm.
Not everyone wants to live in a small prairie city in the middle of Canada.
Yeah.
So, you know, you do have to convince people maybe a little bit to come
Yeah.
and stay. But I tend to find that when people I try to have people come out and meet with me in person when I'm recruiting new people.
Yeah.
And once they see what we have, then I find people are pretty willing to make the journey and stay out here.
I think you make, I think it's a great point that you make, really coming, having people come out and see. And
Hmm.
I think that's important because I've never been to Saskatchewan or to Saskatoon. So in my mind, it's a couple of things that I've seen on TV, but that's
Sure.
about it. And I
Yeah.
think it goes back to some of the advice for the trainees who will be listening to this episode, is really just go out there and meet the person, meet the
Hmm.
lab head, meet the people in the lab, but also
Yeah.
look around in the city, in the university, and you never know
Hmm.
what you'll find, and if you're gonna like what you
Yeah.
see.
Absolutely. And
And
that's
I
great
think,
advice.
thank you, and I think it's also a great thing to have all the toys, all the tools that you need, and have them also available, and have
Mhm.
the support from you and everybody at the university or in the department and in the lab to advance the research.
Yeah.
You mentioned wearing multiple hats.
Hmm.
How do you get it done? Like, how do you juggle all of these?
Yeah. Um, so I put up a lot of little systems in my day-to-day life to help
Mhm.
me manage and keep organized. So my calendar is planned down to the minute for at least Monday to Friday. And that includes scheduling and balancing family time. So for my time with my partner is slotted as a time slot in my calendar, which sounds kind of sad and formal, but it's really helpful. And, this evening I'll be taking my son to one of his sports programs, and that's slotted in my calendar as a specific thing that we're going to do tonight. So everything is planned that way. And then just systems for organization and computer folders and shared drives and everything. I find for me, hyper organization is really the only way that I can survive.
I
And
love it.
sometimes that means scheduling relaxing time, right?
Well, it's interesting because I'm going through a similar exercise. I recently listened to part of the book called Get Things Done.
Okay.
And I didn't have the patience to listen to the whole book. Instead of I listened to the presentation to kind of understand how do you get organized.
Hmm.
I work very well on lists and things like that, but I'm running so many projects at the same time that at some point I need to unburden my brain and I need to prioritize, and I need to prioritize based on different aspects, not only on deadlines, but also on the time I have to work on something. And I
Hmm.
love that you just mentioned that everything is timed down to a minute, and even relaxing time, and even your time with your kids, with your partner. And I think it's amazing. And I think a lot of people listening should think about, one, when it comes to their careers, what do they want to do? What's the end goal? And also think about a system that works for them to reach that goal. I think
Yeah.
it's part of that, that those tools that you need to develop and figure out which ones work for you in order for you to reach your goals.
Yeah, I completely agree. And I tell my people in my group, here's what works for me. This is my system. I'll show them my calendar and all of its nuttiness, and it's color coded and everything. And I'll say, this is what works for me. You feel free to try it out, but I'm not saying that you must use this system, but you need for yourself to develop a system that's going to
Yeah.
work.
Yeah.
Yeah.
And I think that's something that I could have benefited from, for example, during my PhD or my master's, because when you're in the lab, it's go, 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 do this experiment, read that paper, do this presentation. And I never took the time to stop, build the system,
Hmm.
or at least not necessarily not halt everything, but take the time to slowly build out the system and then make it easier for me to run these experiments and do all of those things together. So I think it's really interesting and important what you're saying. When did you develop the system that works for you?
During my PhD. Um, so I joke that I have a child per degree. I had a child born during my bachelor's. I had a child born during my master's. And during my PhD, I really felt like I was starting to fray around the edges. Like I'd open up my documents folder on my computer, and it's just like a wall of files, right? And I felt that fraying in my mind start. And I think it was around the time I did my comprehensive. And I don't know how it was for you at your institution, but for us at Dow, the comprehensive was like you unplug from lab work
Hmm.
and you focus on writing. And I used some of that time selfishly to like reorganize myself.
That's amazing. Yeah, we didn't have that, but I took it anyway because I can't. I'm the type of person who cannot be in the lab, run experiments, and write.
Yeah,
I need time to disconnect and not have to run for my Western blot or red plate or you name it.
absolutely.
That I think also goes to figuring out what works for you
Mhm, mhm.
tools wise. Let's go back to science a little bit. I'm really interested
Sure.
more about the projects that you're working on in the lab,
Mhm,
and what kind of tools are you using to answer the questions you're asking around the CB1, and you mentioned orexin as well.
mhm. Yeah, so for CB1 and CB2 and orexin and 5-HT2A, for a lot of our in vitro assays, we're using high throughput screening based systems.
Mhm.
So for the cannabinoid receptors, we've started using a lot of the enzyme fragmentation complement assays,
Yeah.
like the DiscoverX type assays, or for the calcium assays, it's an Aquiozen based
Mhm.
assay. But we find those really work well as initial systems for screening. And I find too, for particularly for master students or young students, having to be able to just go through a step-by-step process really helps give them confidence. Oh, I can do this. I can get data. I can.
Yeah.
And then from there, we're using immunohistochemical approaches to look at things like ERK phosphorylation, and old school radioligand binding assays to look for, just binding to
Yeah.
GPCRs. And then we have others in the lab that are primarily focused on animal work with mice or with rats. And for that, it's, some basic kind of IP injection studies of compounds looking for early physiological changes. But then we've also started some new cannabinoid based routes of administration that I'm really excited about. So during the pandemic, we purchased smoke chambers so that we can expose our rodents to inhaled cannabinoid smoke rather than injecting them. Because I don't know about you, but I don't know a single human being that injects cannabinoid.
Yeah, no, I don't either. I don't
So
either.
So we've been using that as a newer model for exposure, and it's been giving some really interesting results. So
Interesting. And I think it's less invasive as well. I mean, the
yes.
stress that the animals go through, and it pretty much goes to what you were mentioning about animal welfare and ethics
Mhm.
around using animals in research. Yeah, I
Yeah.
can think about edibles and also inhaling, but injecting is definitely not something that
Yeah.
happens under out there.
Normal circumstances.
Yeah,
Yeah.
I
It
know.
doesn't
Yeah.
happen. It doesn't happen in the wild, if you will.
Yes, definitely not in the wild. I wanted to also ask, so anything you can share with us related to, some of the latest discoveries. Obviously, if it's published, please feel
Mhm.
free to. If it's unpublished, I'll totally understand
Sure,
the
sure.
need for not talking about it. But what have you been? What are the new things that you've been looking at and you've discovered in the cannabinoid field that maybe you found you weren't expecting those results?
Yeah, for sure. So first, I'm not a person who really cares at all whether it's been published or unpublished. Like I'll tell you whether or not it's published or that it's still a work in progress. But I'm never going to be a person that like holds it close to his
That's
chest.
great. It's not my personality either.
Yeah. So for the cannabinoids, the kind of latest and greatest that we've been working on is a rodent model of cannabis exposure in utero. So in this model, we're using those smoke chambers, and we're exposing pregnant rat mums. We call them dams. We expose the dams to cannabis smoke, and then we're tracking the babies
Mhm.
through development. So this is unpublished work, very much still in progress. And we initially started with 5 groups, if I'm getting that right. So we had an air control group, a CBD smoke group, a THC smoke group, a CBD injection control, so we could compare to the inhalation, and a THC injection control. Honestly, we included the CBD groups as to kind of like put fears from the reviewers aside, because the reviewers from our grants, they were like, well, what about CBD, and we were kind of like, well, CBD won't do anything, it's fine. CBD does things, and it's
Interesting.
been quite a surprise to us, but consistently in our CBD injection groups and in our CBD smoke groups, we see smaller pup sizes. They're slower to get back to their mums. They have more resorptions in the mums, which is indicative of like a loss during pregnancy. And the mums themselves aren't gaining as much weight during pregnancy. We've also seen
Interesting.
a few kind of like craniofacial abnormalities, but those are far less consistent, and we need a lot more. We need a lot more work to understand what's happening there. But yeah, we included CBD as a control. We don't know how it's doing these things, but it's definitely doing something, and it's not benign.
So interesting because, well, in Canada, cannabis is legal. It
Yep.
is legal in some states in the US as well.
Mm.
And you just type in, CBD, or you type in THC or edibles in Google, and you have all of these products that come
Yeah.
out that you don't have, no one has a lab at home to test, the purity or the
Mm.
quantity of whatever is on the label. I don't think it's regulated. So it's an interesting piece of work that should, make people think about what do you ingest during
Yeah.
pregnancy.
Right. So that's the cannabinoid piece for the orexin system. My very talented PhD student Jay, she'll be leaving me soon, and I'm very sad to see her go. But it's good problems. Um, she's been working on the orexin system, and when she started her work with studying some of these orexin receptor antagonists, she was noticing a lot more variance in her female animals compared to her males. And she's very astute, she's very good at making these observations. She said, I'd like to go back and retest animals based on their phase of their estrus cycle. And we actually are now seeing differences in estrus cycle response, particularly in the nociceptive or pain sensing pathways, depending on what phase of the estrus cycle the females are in. And some of that work we've published, some of it it's got more work to do. It's ongoing, but it's fascinating, and has all sorts of implications in terms of how we study within sex differences and between sex differences. So
I love it, and I really I've always been fascinated whenever I've seen um, animal studies. The questions I had is, did you do this in males? Did you do this in females?
yeah.
And what was their age?
Mm-hmm,
Because the
mm-hmm.
tendency is to do this potentially more in males and do them young.
Yep.
But I'm glad that you're taking in these things into consideration because it's important. And this may be expanded to studying other compounds, other
Mm-hmm.
GPCRs, other systems. And I'm pretty sure there will be surprises there.
Yeah, and I just appreciate it because, again, like highlighting the strengths of the folks that I get to work with. Jay made a very astute observation, and she followed up on that, and I think that's just awesome.
I think so too. And I think it's also awesome that, she had the space to make that observation and the support to be told, yes, go, do
Mhm.
it. Let's see what happens. All right. So I always ask this question, and I think the answer is pretty much actually 100% yes. But I always ask the guests to kind of expand on it as to why they think the answer is yes to the question, do you think GPCRs are still good drug targets?
Oh yeah, absolutely. Um, so there's so much more to be discovered. I've been following the allosteric literature on GPCRs pretty closely. And even within that, there were some really cool studies recently to look at. I think one paper called it the pocketome, which is like all the sites on the receptor that we don't think about, but are still druggable targets, right? And so you have all these different pockets, like a bad pair of cargo pants that you can put things in. And there's thousands of PhD theses just within that one idea alone. Um, yeah, so there's so much more to do.
I love it. Pocketomes. I'm gonna steal this. I think I really
That's not
love the
mine.
idea.
I can't remember who published on that, but it
I'm gonna
was
look it
really
up
good.
and definitely give them credit whenever I'm
Yeah.
presenting. But I think it's a great, we have the signalosome and the proteasome and the you name it. Man, we have a pocketome. It's awesome.
Mhm, mhm.
I love it.
Yeah.
You mentioned at the beginning that you got it when you got interested in the cannabinoid receptors. You were looking at what other tools are available to study other GPCRs, and you use those to study the cannabinoid system.
Mhm.
Knowing what we know about the cannabinoids, knowing what tools we have to study the cannabinoid system, what else do you think is needed? If you had a magic wand to come up with a new tool that
Yeah.
would help us, move the field forward, what would that look like?
So I think one of the things that would be amazing to see would be specifically fluorescent or traceable ligands,
Mhm.
so that we could target the receptor or different receptor subtypes more specifically. I know that there are a couple of groups out there that have started down this track and they're interested in this.
Yeah.
But historically, with a lot of GPCRs, cannabinoid receptors are no exception. The antibodies, they aren't good. And so it would be lovely to have something like a specific ligand that targeted, say, CB2 versus CB1 that also fluoresced that you could use as a detection agent. That would be my dream. If someone could make that for me and send it to me in the mail, I'd be very happy.
Awesome. Actually, there is a company in Spain who
Mm-hmm.
specializes. I don't know if you've heard of them. They're called Celtaris. I'll send you
Okay.
the link, and they specialize in making fluorescent GPCR ligands.
Yeah, and I think they might have presented that group might have presented some preliminary work at a past cannabinoid meeting saying they were beginning to explore this, but
Yeah.
yeah,
But it's a tricky because of the nature of the ligands.
absolutely.
I think we've come a long way when it comes to chemistry and labeling and all of these tools that we have. So hopefully,
Yeah.
fluorescent cannabinoid ligands are just around the corner.
Mhm, mhm.
We've talked a little bit about advice for junior scientists who want to contribute to the field in general. Anything else you want to add?
About junior scientists
Yeah, about
specifically?
yes, well, scientists specifically, but yeah, junior scientists, PhDs, and
Yeah.
postdocs who want to contribute to the field.
Yeah, so I would say, the biggest positive experience for me was getting out to meetings or conferences or being able to talk with people
Mhm.
and just meet and engage with others, whether that's on Zoom, like we're doing right now, or in person. I think that had the biggest impact on my career. And then the other thing, a lot of times grad school and training, it can be really isolating. And just not to forget that you're part of a team, you're not alone, and there's always people to reach out to and talk to. Maybe that's your advisor, maybe it's a member of your committee, maybe it's someone else in a totally different lab. Um,
Maybe it's you.
Maybe it's me. It could be me. But it takes a village, I think, and it still takes a village. One of the things I didn't mention earlier, but I loved for early career researchers in their new faculty position is I had an advisory committee when I started as a faculty member.
Mhm.
And I started my job and my dean said, "Hey, you're gonna have a committee just like you were a grad student." And initially I was like, "Why? I'm a big boy now. Why do I need this?" Um, but it turned out to be super helpful. And I would say for early career researchers, if you can make that happen and get some senior researchers on your team, it makes a huge difference.
It does. It does. And I think, they've gone through the experience, they've seen it all,
Hmm.
and having multiple people who've gone through similar yet different experience that the same things differently
Yeah.
can be very valuable. How would you go about finding a mentor or finding these people that you're talking about that could help early career scientists and junior scientists support them in their journey?
Yeah, I think I would start kind of at my local level, like within my institution, find people with shared interests. And this might sound counterintuitive, but get to know those people personally is just as important as getting to know those people professionally, right? The people that I have the most success collaborating with are the people that I can get along with and go and have a drink with after work. So I would say talk to people at your own institution and see kind of who has shared interests with you. And if you see someone online, maybe they're through Twitter or through another social media,
The
I would,
ecosystem. I'm
yeah,
gonna plug it in.
yeah, great. Yeah, that's a great idea. Cold call people, you know. One of my kind of recent really successful collaborations was because one of my former PhD students, Asher Brandt, he cold called a researcher at the University of Ghent, Christoph Stove, and was like, "I like what you're working on. I have some ideas," and it's turned out to be very fruitful. So,
I
yeah,
think you make a great point about, cold calling and not being afraid to just step out and ask because the worst that can happen is you're not going to get an answer or they're going to say no. But
hmm,
most of the time I think we live in an era and in a field that's very, helpful and people want to talk and people want to collaborate.
yeah,
And
yeah,
that's really important to keep in mind whenever as an early career scientist or as a junior scientist you're looking to interact with people and you never know. I love
yeah,
that you mentioned, having your student pick up the phone and talk to this other scientist at the other end of the world when you think about
mhm,
it,
yeah.
and it turned out to be a fruitful collaboration.
Hmm, hmm.
All right, two last questions because I know you have a hard stop. Top three aha moments that defined your career as a scientist.
Tricky one. Um, Doing experimental design at Agriculture Canada, like randomized complete block design for canola treatment crops. I don't know, for some reason that really scratched an itch in my brain, and I was like, oh, I like this. Let's do more of this. So that was maybe the first one. The second one would be when I finally figured out and understood how nonlinear regressions worked and like how curve fitting worked.
Yeah.
That was kind of like a big light bulb moment for me. And then I guess most recently seeing like the pure joy on one of my students' faces when they were successful with one of their own grant applications. I don't think I've ever felt so internally happy, like almost on the level of a child being born. And not quite there, but it's pretty close. And so those would be the three that immediately come to mind.
That is awesome. I think you're the first and only guest so far who had this expressed this, in their top 3 aha moment, being proud and happy to see their students. Not that others haven't, but I
No, no.
think I love it. All right, last question, much easier than the previous one. Where can people find you in whenever you have job openings or in
My
general?
Twitter, yeah,
Yeah.
my Twitter at RB Laprairie, or on my campus website. So you can just look at Robert Laprairie at Pharmacy and Nutrition. Those are the easiest places to find me.
Perfect. So take home messages, reach out to Robert, go visit Saskatoon, and who knows, maybe you just found your new home.
Yeah, absolutely. It's a dry cold. It's fine.
I think that's also a great point to make, that it's a dry cold. I think anything that's humid is more dangerous, heat or dry,
Mhm.
than the dry cold. All right. Thank you so much, Robert, for your time. I really enjoyed our chat. Really enjoyed learning about you and the work that you do in the lab, and hope to keep in touch.
Thank you so much. And I really enjoyed this too. It's been great.
Thank you. Thank you for listening to this Dr. GPCR podcast episode. We'd like to thank our guest and our Dr. GPCR team, namely Attila Forrest, Ines Pinero, Montserrat Avila Zozoya, and Nipuna Wirasingo. A huge thank you to our ecosystem partners for their support: Domain Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, and Montana Molecular. You can connect with our partners directly in the ecosystem. You can also connect with our Dr. GPCR team directly in the ecosystem as well. Join us today. Also, please subscribe to the Dr. GPCR newsletter, find us on YouTube, and if you like our podcast, leave us a 5-star review on Apple Podcasts. You can also leave us a testimonial at drgpcr.com/testimonials. And another great way to support us is to share your favorite Dr. GPCR program with your network and colleagues. You can email us at any time with any questions or suggestions at hello@drgpcr.com. Until next time, stay safe.